Boa noite a todos. Estamos aqui para a nossa última live, nossa última apresentação nesse webinar sobre qualidade de carne bovina. E é com enorme prazer que nós vamos fechar esse evento com chave de ouro, com o Dr. Chris Calkins, falando sobre propriedades tecnológicas da carne. Meu nome é Ana Maria Bride, sou tutora e coordenadora do grupo de pesquisa e análise de carnes da Universidade Estadual de Londrina, e eu queria aproveitar essa ocasião, já que é o nosso último encontro, para agradecer a minha equipe, os alunos que compõem o GPAC, que organizaram. Eu, na verdade, estou aqui mostrando o meu rosto como o nome, representando o grupo, mas quem realmente trabalhou foi quem está nos bastidores aqui atrás. Então, eu queria agradecer os alunos de doutorado, Amanda Gobete Barro, a Evelyn Rangel dos Santos, o Guilherme Agostines Ferreira e a Jéssica Gonçalves Vero. E também os meus alunos, meus orientados de mestrado, a Daniela Kaiser Terto, a Edmara Rodrigues Correia e o Victor Furlan. Então, foram eles que realmente colocaram a mão na massa ou a mão na carne e tornaram esse webinar possível. Então, queria deixar aqui público meus agradecimentos a essa turma que trabalhou arduamente para colocar no ar. Gostaria de chamar agora a nossa hoster, a Luiene Moura Rocha. Boa noite, Luiene.
Boa noite, professora. Como vai a senhora?
A Luiene é minha ex-orientada de mestrado, fez o mestrado em ciência animal na universidade. Depois ela foi fazer o doutorado na Universidade de Laval, no Canadá, e também fez um pós-doutorado na Universidade de Sherbrooke, e hoje ela está trabalhando e morando no Canadá, e ela que vai conduzir, então, esse nosso webinar. Bem-vinda, Luiene, obrigada, quero agradecer a você também por estar nos ajudando nessa tarefa, sua contribuição foi muito importante para a gente poder realizar esse evento. Muito obrigada.
Sou eu que agradeço realmente o convite, para mim foi uma honra ter sido convidada por vocês, e é uma honra estar aqui contribuindo, assim, para o nosso setor de ciência da carne.
Então, pode ficar à vontade para apresentar o Dr. Chris Calkins.
Muito obrigada.
A todos. Obrigada.
Boa noite, professora. Bom, boa noite a todos. Como a Ana Maria Bride já apresentou, eu sou a Luiene. Eu vou estar com vocês aqui essa noite. E, como vocês lembram, eu vou falar agora um pouquinho em inglês, porque eu vou apresentar e vou chamar o Dr. Calkins. Então, good night, everybody. So here we are for the last presentation of this webinar of beef quality. So I'd like to welcome everyone. So thank you very much for being here with us today. And just to remember you that this amazing webinar was organized by the Carcass and Meat Quality Evaluation Research Group, known as the GPAC, from the Universidade Estadual de Londrina. The GPAC was created in 2006 by Dr. Ana Maria Bride and is currently composed of 15 undergraduate students and 7 graduate students in animal science and medicine veterinary from the Universidade Estadual de Londrina. The GPAC, they work in a close collaboration with the industry, and they do research in the field of pork and beef quality, and as well on fish meat quality. So I would like also to invite you to take a few minutes to join us and follow the GPAC Instagram to know a little bit more about the research they do and the results they obtain. And so it's going to be really interesting to have you there. So please, if you can, they are going to put the link so you can join us to follow them. And just to talk a little bit about the well, the Universidade Estadual de Londrina is located in the south of Brazil, in the state of Paraná, which is a big producer of poultry and pork meat. And today, our special guest is Dr. Chris Calkins, which is going to offer us a live presentation. So today, it's not a registered presentation. Today, he's going to be live for the presentation and also for the questions and answers section. So he's going to talk about the physicochemical and sensory characteristics of beef. So before start the presentation, I would like to introduce you Dr. Chris Calkins. So good night, Dr. Calkins. How are you doing?
I'm doing great. I'm excited to be here, and I'm very pleased to be invited to be involved with the webinar.
So it's a pleasure to have you here with us, and I'm going to introduce your CV. So before you start the presentation, because it's a really great pleasure to have you here with us. So it's a really good pleasure to finish this webinar with you. So Dr. Chris Calkins, he has a bachelor degree in animal science from the University of Texas, A&M. He has a master's degree in food science, food technology and science from the University of Tennessee. He has a PhD in animal science from the University of Texas, A&M. He is also currently professor at the University of Nebraska. He's a member of the American Meat Science Association. He's also a member of the Institute of Food Technologies and the American Society of Animal Science. Dr. Calkins have received more than 30 honors and awards. So I'm going to tell just 3. In 2014, he has received it from the American Society of Animal Science in an Animal Industry Service Award. In 2011, he received the Meat Research Award of the American Society of Animal Science. And also in 2011, he received the Distinguished Research Award from the American Meat Science. So he also has numerous scientific publications related to the characterization of muscle properties, the sensory characteristics of beef, and the effect of production system of palatability of beef. So please join me to thank Dr. Calkins for being here with us. So Dr. Calkins, do you want to tell something before I start?
Just happy to be here. I'd say let's get rolling.
Okay, so the floor is yours. So let's start the presentation, and thank you very much again.
Thank you. It is a delight to be here. And when I first was invited, I want to thank Dr. Beachy for the invitation. I asked questions about the title that we had been given. And one of the things she said was not only to talk about those characteristics, but also the possibility of predicting and being able to forecast what those characteristics are in meat. I happen to think that's a very important and fascinating area, so I'm delighted to address that particular question today. As I do so, one of the technologies that I discuss, particularly related to tenderness, is actually developed by a company that I am affiliated with. As a result, the university requires that I make a disclaimer and that I actually indicate to you that I have a significant interest in that company, both TenderSpec as well as Goldfinch Solutions. And in order to avoid any potential appearance of a conflict of interest, then the university asks that I give you this slide to show you that that situation does in fact exist. I hope by the time we finish the presentation, you will understand that my statements are based on fact and based on science and not biased. But nevertheless, I have to make that clarification right up front. As we talk about the critical properties of meat, particularly those that influence value, today I'm going to focus on 4 specific characteristics. And I know that you know something about each one of these. I will review a few key points, but mostly this is a great opportunity, I think, to remind ourselves that the way we collect data, the way we interpret and present data can have an impact on how people understand the research that we have done. So let's start first with color. And of course, what you notice on the screen right away is this particular steak has some problems with color. There is discoloration. And I don't think we need a lot of research data to know that when the meat starts to turn brown in the retail case, that consumers do not want to buy that product. In fact, Hood and Reardon, way back in 1973, identified that 20% discoloration is enough to cause a 50% reduction in sales of a particular cut. So for me, that's a very important threshold for us to think about. And I really want to focus for a few minutes here and get you to think about the importance of thresholds as we report and discuss data. And so this happens to be some recent data that we collected, data collected during retail display of cuts that were both wet-aged or dry-aged, as well as some wet-aged and dry-aged dark cutters. Dark cutters have a high pH and a very dark color. We'll talk about those in a few minutes. And what we did was on each day of retail display, we identified which products had the highest discoloration, which is a useful way to talk about the various treatments. But what I think we need to try and do is to put that information in context in a way that it means something to people. And so if we use that 20% threshold, now you can see that the the dry-aged products discolored after 4 days of retail display. And we got 2 more days of retail display with wet aging. And so by using this color threshold, it helps us understand the importance of the results that we found. Of course, it's very common to report color using the color space, L star, A star, B star, lightness, red to green, blue to yellow, and we typically report each of those characteristics individually. One of the previous speakers, Dr. Hopkins, was co-author on a paper that pointed out that when that A star, that redness value, reaches 14.5 or better, that is an acceptable color with consumers. If it drops below 14.5, the color is no longer acceptable to a group of our customers. And so again, the same characteristics, the same samples I described earlier. In this case, I've shown A star. You can see that in the case of the wet age, they had more redness that declined during retail display. That was also true for the dry age products. But what's meaningful for me is that threshold again. In this case, you can see that in the case of the dry age products, they reached their maximum at about 3.5 days, whereas the wet age products were able to last 2 additional days. And regarding A star, none of the dark cutters were red enough to meet that threshold. Finally, when we talk about the variables in the color space, L star, A star, B star, we get 3 values, which makes it really hard to conceptualize and think about what the color actually is. And what's especially challenging is when we want to compare colors. A color might be different in L, but typically it's different in L and in the A dimension and in the B dimension. And so we have all 3 coordinates, making it a challenge to compare one to the other. Fortunately, there is a characteristic called delta E that is the relative distance between the 2 points within the color space. Again, I think that's a meaningful term when we describe color of products and when we start to characterize and evaluate color prediction as well. In this particular case, there have been 2 presentations, one by Zhang et al. In 2003 that notes that a significant change when you have 10 units of difference in delta E. And another group has published that even at 2 degrees, a value of 2 for delta E is sufficient for a standard observer to recognize the difference between samples. So now we get an understanding of the magnitude versus the actual meaning of the absolute values that we're looking at. Of course, we know, and we always tend to talk about color. Everybody gets excited talking about the chemical state of pigments. I too will talk just a little bit about that today. But before we get there, I want to remind us that we know there are other things that also impact color. One of those is muscle structure. And that's because part of what we perceive as color is based on the reflectance of light from the surface of a sample. But of course, the surface is not smooth as glass, as showing in this figure, but sometimes the surface is rough. The texture is different, and we get the scattering of light, more of a diffusion of light. And that impacts the color perception that we see. So as an example, the carcass on the left is a dark cutter, high pH. We know that as a result of that high pH, the muscle structure is different and the reflection of light is different. Of course, that pH also impacts mitochondrial function and the chemical state of pigments as well. But we can't forget that there's this fundamental structural impact that we should keep in mind when we both measure and predict color. Secondly, of course, is the amount of pigment. Again, we tend to jump into the chemical state, but just using beef for an example, you can see that going from veal to young beef, we can see a doubling or even tripling of myoglobin concentration, and then going to mature beef, even a doubling after that. So animal age also impacts color. We also know there are differences from one species to the next. Beef is darker than pork, is darker than poultry, and so on. So we have those inherent animal differences that we should keep in mind. Finally, then, the chemical state of pigments. We know what this is. I won't review it in great detail, except I want to, again, call out for you one way to think about this product and this particular property, because I think it's meaningful in understanding the values that we collect. We know that when you very first cut a steak, it's in the deoxymyoglobin state in the upper left-hand corner of the screen. You kind of have a little purplish color to it. When we allow oxygen to join, it blooms, it oxygenates. We get that bright red color, which we see on the right. And then if we leave it long enough in the retail case, the steak will eventually turn brown. What you should remember and think about is that we're talking about a population of myoglobin molecules, not just one. And so at any given time, we don't have 100% deoxy, 100% oxy, or 100% met. We have a relative distribution between those. And we can visually see that when we look. That's why I like the discoloration measure evaluated by a panelist as a way to indicate what color actually is. But in fact, the myoglobin molecule itself can go around this circle, as you see, and color can change depending on the situation and the conditions that we have displayed the meat. Now, if I was teaching a class, I would stop and ask you, what are these equations that I have presented in front of you? Well, you probably already recognize that Krzyzewski in 1979 in the journal Meat Science really defined the equations to calculate the percentage of the pigments that are in the met myoglobin, the myoglobin or deoxymyoglobin, and then the oxymyoglobin states. And notice that we're talking about absorbance or reflection at specific wavelengths in these equations. And I just want to call your attention that every one of these equations has 730 in that equation. Now, why is that? It's because we know that at 730, we actually get some insight into the muscle texture as well as the characteristics of the chemical state of the pigment itself. So this is a complex slide. I won't go into great detail. I will tell you that you can go onto the web at Minolta, and they have published a little booklet called Precise Color Measurement. It is a wonderful presentation understanding color and color measurement and how we present it. But there's a particular point and a particular reason why I put this on the screen. First of all, you note that the eye can perceive red, green, and blue because the eye has sensors that are sensitive to those things. And it's been learned that in the visible region, roughly 400 to 700 nanometers, in the visible wavelength, there is a collection of wavelengths that contribute to our perception of blue, and a different population for green and an even different one for red. You can think of that as colors being defined in those three characteristics Those are called tri-stimulus values. It's how our eyes work. Now, my main point is that we often use a colorimeter to measure color. The colorimeter has 3 sensors, one for red, one for green, and one for blue. And so the colorimeter is helpful in defining color, just like in that color space that we described earlier. The challenge, however, is that the colorimeter will also give a spectral reflectance curve. And many people like to use those values to do the calculations from Krizwicki, like I just showed a moment ago. Notice, though, on the bottom of this screen, there's a representation of what happens when you use a spectrophotometer rather than a colorimeter. And when you use a spectrophotometer, you actually get a spectral graph. You, in essence, get readings at every nanometer or every couple of nanometers along the wavelength. Now, the reason that's important is that colorimeter has a 10 nanometer bandwidth. So it's a very broad measure, whereas when you use a spectrophotometer, it's a very narrow measure. I'd like to explain why that's important to us. This is an example of reflectance, and you can see we have deoxy, oxy, and myoglobin presented on the screen. And there's this wonderful point at 525 where the contribution of all 3 pigments to the reflectance or the absorbance value is equal. And so back on those Krizwicki equations, 525 is included in all of those. So you can see the importance of using and measuring reflectance or absorbance at precisely 525. But what if we're off 10 nanometers? Now when you look, the contribution of myoglobin is about 50% higher than the contribution of oxymyoglobin or deoxymyoglobin. And so that variation, using a colorimeter to give us spectral results, has some limitations, and we need to be very careful when we interpret color because of that. Now, the other thing I just mentioned here, and we'll go into this a little bit later, is that when we think about color, we always think about that visible portion and think of all of the different nuances of the color characteristic that we can find when we look at the visible wavelength. But just to the right of that is the infrared, and between the two would be the near infrared region. If we could see into that region, we have the potential of seeing a lot more information on the meat than what we can visibly see with our eyes. That will become important in a few minutes. So it's color that we're wrapping up here. And now let's talk a bit about marbling. Now, you could argue that marbling is not a physicochemical characteristic. It does represent how much intramuscular fat, how much fat inside the muscle is present within the product. However, because marbling is related to the characteristics, particularly tenderness and flavor, and also juiciness, then I think it's important that we acknowledge and recognize the contribution of marbling to those things. And so this is just a plot that shows that as marbling decreases from the left side of the screen down to the right side of the screen, then flavor desirability also decreases. This, of course, is country specific. These data were collected here in the United States. We like the taste of marbling. We find that desirable. So the more marbling, the better for our system. That may not be true in other countries of the world. I want to tell you about a little study we did. We were curious about how people evaluated marbling. And so we showed people 2 steaks similar to the 2 marbling levels you saw on the screen. And we simply asked, which of these 2 would you buy and why? In 2 different cities, first in Chicago, 85% of the customers said we would pick the low marbled steak. And in San Francisco, 2/3 of them said the same thing. Now, I just told you that we like the taste of fat, we like the taste of marbling, and yet you can see the majority of our customers are downgrading or selecting against the marbling. That's an interesting perception, and we needed to know more. So that question of why did you make that choice is answered on this screen. The red bars are the answers that customers identified as to reasons why they like the high marbled steak, and the white bars for why they like the low marbled steak. You can see that those who selected and said, I like the one with a lot of marbling, actually recognized it was marbling. They understood it contributed to flavor, juiciness, and tenderness. Whereas the people that wanted the leaner, the one with less fat, were more interested in the negative aspects of fat, feeling like fat was too high in that steak, and therefore it was somehow unhealthy. Interestingly, even in the high marbled steak, it was less than 10% fat. Now I want to go on and talk a little bit about flavor. And I want to tell you about a research project we did, actually a series of these. And one of the first questions we wanted to answer was, how valuable, how important is flavor if you remove the effect of tenderness? So if you made sure they were all the same tenderness and flavor was the only difference between the sample, what impact would that have? And in the first study, we looked at marbling. And in the second study, we compared domestic grain-fed versus an imported grass-fed product. So quite different flavor characteristics. What we did was we went to the cities when the panelists came in. We actually handed them cash for their participation. Cost a little more in San Francisco to have them come to the panel. But we gave them $25 to $35. And they had that money in their hand. Now, what we did was we gave them 2 samples, and they rated them for sensory. And after giving them their sensory ratings, then they were allowed to bid, in other words, like an auction, write down on a piece of paper, how much would you pay for one of these steaks? And what's most important is not necessarily how much they would pay for one versus the other, but what we were most interested in is how much more would you pay for one than the other? So it was real food, real money. If they won the auction, they in fact had to buy the steak. So we did everything we could to find out what was their genuine, true feeling for which steak was more desirable. In the first comparison, we compared upper 2/3 choice marbling to select, like you saw in that photograph a minute ago. And what we did was we screened a lot of loins, so they all were the same shear force within 0.01 kilograms. So the tenderness was not different, only the flavor due to marbling. Of course, in this case, they didn't see the steaks, they only tasted the steaks. And here's what we found, was that tenderness was rated the same, but there was a distinct flavor advantage for the high marbled steak compared to the low marbled steak, and also a juiciness advantage, and therefore overall acceptability. So they were able to taste the fact that those high marbled steaks were in fact juicier and overall more desirable to them because they like the flavor. Here's the interesting thing. When we asked them how much would they bid, well, you can see a number of them were indifferent. But when they had a preference, 2 to 1, they preferred the high marbled steak, and they were willing to pay $1.30 per pound more for a product that had the high marbling. They're willing to pay for their preference. And those that liked the leaner product, they too were willing to pay what works out to be about $3 a kilo more for the one that tasted the way they want. Well, surely flavor is important, and we should think about that as we try and evaluate and predict characteristics. Now, I told you we also compared grain-fed and grass-fed. The grain-fed was domestic, very lean, select grade, which would have been slight level of marbling, aged for 9 days. And then we compared it to an imported product, which also had slight marbling, which aged for about 30 days. So you can imagine the taste of these was too different. Once again, we measured a lot of loins, made sure that the shear force was exactly the same. This is a picture of one of the imported loins, aged for 30 days. You can see a great deal of package integrity. It was a high quality package, a high quality product. I'll be honest with you, this came from our friends in Argentina. And in case anybody wants to know, according to them, it's the best beef in the world. I'm not sure everybody in the United States would agree with that statement, but at least that's what they were saying. So it was an all-natural product, grass-fed, aged for 30 days, compared to a similar amount of marbling and a similar tenderness. And in this case, not only did we see this enormous difference in flavor, but even though tenderness was the same, because it tasted so good, consumers perceived that product as being more tender. They also noted that it was more tender and rated it more acceptable overall. And when it came to the price differential, again, $1.60 a pound for the one that they preferred if it was domestic, and $1.36 if they preferred the Argentine product. I can tell you that overall, over 80% of the consumers preferred our domestic product to the imported product, even though they didn't know it was imported, they did not know it was Argentine, totally based on taste. My point here is flavor is important. It's a critical sensory characteristic. Now let's move on to tenderness. I like to say if you want to entertain a meat scientist, ask them about tenderness. We've been studying this for 100 years, and so we know a lot about tenderness. And in the United States, we pay for tenderness. So that steak in the middle on the top, that is a tenderloin steak, a filet. You can see that it is the most expensive steak that we sell. We have a ribeye over here on the left, a strip steak. Those are both longissimus muscles, very high value. Whereas when we get over on the right-hand side, a round steak from the hind limb, this particular one would be part of the adductor, a much lower value. So in our market, we recognize and value tenderness. And consumers can tell a difference. Research is published has shown that they can detect as much as a ½ kilogram difference in Warner-Bratzler shear force. So we want to pay attention to that. And it is important in the United States. We do nationally, there's an organization that does a beef quality audit about every 5 years. And for the last 4 or 5 years, tenderness has been identified as one of the top quality concerns in that beef quality audit. People are very sensitive to tenderness. In addition to that, that same organization conducts a survey and they ask people, does beef taste great? So this is about flavor. And you can see 8 to 14% would disagree. All the rest would say, yeah, beef tastes great. But when they ask the question, is beef always tender? A third of our customers say, no, it's not. So tenderness is important to them. And in fact, if you look in the literature, you will see that people are willing to pay anywhere from $2 to $5 more per kilo for a steak that you can guarantee is tender. There's a lot of incentive to focus and try and predict tenderness. A friend of mine, an economist, Glenn Tonser at Kansas State University, summarized research a few years ago and concluded that just in the US alone, if you could guarantee tenderness, the market would be worth $760 million. And a further study that he did indicated that on average, consumers would be willing to pay an additional 30% for guaranteed tender beef. So we think there's lots of incentive to focus on this particular trait, this particular characteristic. Now, there are a number of things that are related to tenderness. We know many of these already. We talked about marbling. This is an interesting study published in 2013. And in this case, the trained sensory panel evaluated a wide variety of marbling scores. And those marbling scores, the more marbling, the lower the Warner-Bratzler shear force, and the higher the taste panel tenderness rating. So looking at these trained panel results, you would see that there would be a preference for the higher marbling scores. In fact, you can calculate what is the likelihood of a positive experience when consumers eat steaks that have that high marbling. And what you see is the prime steaks, the highest quality grade, the highest marbling score we have, a 98 to 99% chance of satisfaction. When we go to the upper 2/3 of choice, 88 and 82%. And then as we drop down into the lower level of choice grade, which is small marbling, just 60%. And then in the select grade, only 30% of the people found a positive experience. Now, of course, that was with a trained panel, and the research was done approximately 8 years ago. And so there's been some recent work, which I'll talk about in just a moment. I got ahead of myself. So here is their results, in that you can see that 81% of the difference in overall sensory scores could be explained by tenderness. So this would say tenderness is the most important sensory characteristic in terms of overall satisfaction. You can see there's some significant flavor issues here as well. Buttery beef fat flavor is really marbling, and that alone explains 10% of the variation. So that was 8 years ago. Since then, I can tell you that we have dramatically reduced the amount of select grade beef. We have increased the amount of choice and prime beef produced here in the United States. Some would say then, as a result, that we have a very high level of tenderness, and consequently, the importance of tenderness to consumers would be reduced. Tenderness is there, they can check it off, and now their distinction occurs because of flavor. And so this is the results of that study that was published by O'Quinn in 2018. And in this case, you see tenderness and flavor about equal in importance, maybe flavor a little higher, but possibly because most of the products were in fact tender. And then juiciness was important. Now these guys Travis did a really interesting analysis that I wanted to call to your attention. And what he did was he said that if tenderness is acceptable, then you only have about a 10% chance of rejecting the steak. If juiciness is acceptable, about 1 in 9. And if flavor is acceptable, about 1 in 15. So if it's acceptable, you're gonna rate that steak pretty well. But if it's unacceptable, look at this. You have a 7-fold increase in an undesirable sensory rating if tenderness is the problem, and a 12-fold increase if flavor is a problem. So clearly, tenderness and flavor are important. And you can see if there are more than one defect, if tenderness and flavor both are unacceptable, well, you're at 46% chance. And if tenderness, juiciness, and flavor together, you have 89 times more likely to reject that steak. So what's that say is that if it's juiciness, or if it's tenderness, or if it's flavor, if any one of those 3 characteristics are undesirable, then it's going to be an undesirable eating experience. We need to aspire, and our goal should be to hit high in those areas. Now, there are some other characteristics of tenderness I want to just mention very briefly, because they create a challenge for us in how we try and predict these characteristics. So first, let's talk about muscle fibers, and we'll talk briefly about degree of shortening. So you know that a contracted muscle is got more overlap of those protein filaments as identified in this diagram. And in fact, if you look at a micrograph, you can see much more dense proteins in the bottom one, which is contracted compared to the top. That's going to require more shear force and will be less tender. Anything that evaluates tenderness then should have some way or mechanism of evaluating sarcomere length, or get some idea of contraction. Now, the other part about those muscle fibers that are important is how fragile they are. So our national organization, National Cattlemen's Beef Association, did research on aging, and you can see that the response to aging, the improvement in tenderness that occurs when we store a product under refrigeration is different for different muscles. It's different by grade, and it's different for different muscles. So now when we think about tenderness prediction, we got to take all those things into consideration as well. And you know that as we age, those cathepsins, those proteolytic enzymes activate. So you take a little piece of meat, you put it in a blender. 1 day post-mortem, there's a high degree of integrity in those muscle fibers. And 10 days post-mortem, they're very fragile. They'll break apart very easily. So the number one thing you can do if you want to try and improve tenderness is age the product. Now, this also creates some challenges, because if you are making measurements at day 1 or day 3, and you're trying to forecast what tenderness is at day 10 or day 14, then you've got some issues, because you have to be able to anticipate what that enzymatic tenderization response will be. Lastly, I want to mention about connective tissue. That's all this tissue in between the muscle fibers, as you know. And just as a reminder to everyone, what happens with that connective tissue when you heat it, I took the connective tissue off the surface of a steak, and I just threw it in a skillet. And I took a picture every 15 seconds. So this is 0 seconds, 15 seconds in a hot pan, 30, 45, 60. In 1 minute, that connective tissue has tightened up so much that it's very, very difficult to chew. And so naturally, differences in the amount of connective tissue will have an impact on tenderness. And this just shows you that across various muscles, there's a 4-fold difference in collagen content or more from one muscle to the next. So again, a tenderness prediction methodology needs to take into consideration that particular circumstance. We also not only need to think about how much connective tissue is there, which differs by muscle, but we should also think about the solubility. As an animal gets older, that collagen, that connective tissue gets less soluble, and therefore is less tender. And you can see here, amaturity cattle for us, whether the red bars represent one muscle, the blue bars are another. As the animal gets older, solubility goes down, shear force goes up, tenderness goes down. Now, in addition to the challenges of biology and their contribution to variation in tenderness, there's also some differences and challenges associated with tenderness measurement that I'd like you to think about. So first, I'd call out a slice shear force developed by Tommy Wheeler and Steve Shackelford and others at the U.S. Meat Animal Research Center here in Nebraska. In this case, you take a slice of meat and you shear it and you measure the tenderness. And, you know, they published a paper that showed 95% repeatability between multiple steaks, steak taken on the left side of the carcass, on the right. And initially that sounds pretty good, but I would just call attention as an example here, where here's a slice shear force about 13 on the left, on the right-hand side of this carcass, and on the left-hand side, 19, almost 20 kilograms of shear force. And so that's a case where there's this repeatability really creates a challenge. And how do you know what the true measure of tenderness is of a muscle if you can go from 13 to 19 in one carcass? Warner-Bratzler shear is not without some of those same challenges. Again, those same scientists did a study comparing the repeatability of different laboratories around the United States. And if you'll notice the mean shear force for the 50-some samples they had, look, the one on the top is 4.7, and another lab, it was 2.9 kilograms. In our system, that's enough to differ between one being classified as tough, not just intermediate, but tender. And so measurement errors are profound. They're inherent to what we do, but they complicate the issues of how do we evaluate tenderness. Incidentally, they also pointed out the correlation between sensory and shear force is just 0.77. Not too bad, but again, not perfect, and making an absolute distinction or a measure of tenderness a particular challenge. And then finally, of course, we have cooking. And we know that cooking can make a difference depending on how well done you like the steak. And if you just look at muscle fibers, on the left, they're raw. And as we move to the right and endpoint temperature increases, you can see there's just an increased coagulation and increased hardening of those muscle fibers as we cook. When we look at the collagen or connective tissue, we know the same thing, that they coagulate, they shrink, they contract. And so that degree of doneness will impact how consumers perceive tenderness. And finally, if you think about, well, okay, let's just take muscles along the loin. It's one muscle. We'll just sample it from one end to the other. These are some data published in 2000 that show considerable amount of variation from one steak to the next, just side by side. So there are some extra challenges with this. The bottom line is, if you're trying to predict tenderness, it's a little bit like trying to hit a moving target, and that makes for a challenge. So how have we tried to do that? Well, there are those that are published in the literature who have used near-infrared spectroscopy to do so. And you can go online and you can do a literature review, a number of publications on this. When you have a sensor like this that covers the steak, you get a spectral reflectance curve like you see on the screen in this picture. But that reflectance curve reflects or is an indication of one measurement from the whole steak. So we would say you have good spectral sensitivity, but from a spatial standpoint, you don't. It's all or none. Others have used image analysis. And in image analysis, you can get a nice indication of what the entire steak is, but now you have no spectral resolution. This is just over the visible region. And so what we'd really like is to combine spectral sensitivity with image analysis sensitivity. And that's where a technology called hyperspectral imaging comes into play. When you have hyperspectral imaging, you get a spectral reflectance curve on every single pixel in the image. That means that that reflectance curve isn't the average like you would see in that NIR spectroscopy earlier. And in fact, we could use it to pick out the the red squares versus the black squares in the checkerboard. The NIR spectroscopy would just give you one reflectance curve for that. So this is really powerful. It was published a statement that said the goal of developing hyperspectral imaging is really to design multispectral imaging. So what I want to talk about is when you do hyperspectral imaging, you get a reading at almost every nanometer. Well, that's a lot of information, right? Now, buried in those curves are specific key wavelengths that give us particular information. For example, here you can see that at 1365, there's some information about the proteins. At 1408, information about water and so forth. There's some fat. And so by going in and selecting specific wavelengths that you need to measure, then you have the possibility of not having to measure everything, as in hyperspectral imaging, but you can just measure key wavelengths like you could do in multispectral imaging. And just so you know that it's not a numbers game, I will tell you that the characteristics used to do the tenderness predictions have been correlated to a lot of those biological properties we just talked about. Marbling or fat content, sarcomere length, collagen, proteolysis. And so there is, in fact, a direct measurement of the biological characteristics that impact tenderness. Now, this is the results of our one of our studies on hyperspectral or multispectral imaging to predict tenderness. You can see we were 95% successful in identifying whether steaks should land in the tender, intermediate, or tough category. We think that's highly promising. And as I mentioned at the start of this, I am involved with the TenderSpec folks. And I just want to show you, this is what a TenderSpec device looks like. It's able to capture an image on a moving beef carcass, and in a very short period of time, indicate the tenderness classification. And it's a high resolution image. You can see very small flecks of marbling in this particular image. And at the same time, it also has that spectral information on every pixel in the image as well. And so that particular TenderSpec system, it works real time, it's accurate, it's non-destructive, and it can work at grading, in other words, 2 days after slaughter, to forecast what 14-day tenderness will be. In addition, it gives answers within about 5 seconds. It's been tested so far on 3,000 steaks. And again, that 2-day measuring, forecasting tenderness down the road, which is a particular challenge. It's highly accurate, 95% accurate or better, and can also, at the same time it does a tenderness classification, identify what the quality and the yield grade of the system is. Now, the last thing I want to talk about today as I wrap up is an exciting new technology a good friend of mine, Dale Werner, has been developing. Dale is a Cargill endowed professor at Texas Tech University in meat science, and he has studied something called REIMS, which stands for Rapid Evaporative Ionization Mass Spectroscopy. It's quite a mouthful. Now you know why we say REIMS. I'll describe this for you in a minute. I will tell you that I reached out to Dr. Warner, and he shared slides with me that were used at the recent International Congress of Meat Science and Technology. Don't worry, I'm not going to show you all the slides, but I just want to show you a few so that you can understand that here is another up-and-coming predictive technology, which I'm excited about. I think it has a lot of potential, particularly as it relates to flavor. So this is a novel technology, and what it does, it also provides real-time analysis, no special preparation for samples, although there is a little bit of sample destruction, as you'll see in just a moment, and it's a handheld device. In essence, what you have is you have this, it looks like a pencil. They call it an eye knife. And when you draw the knife across a piece of meat, that inductively causes heat, which cauterizes the meat. In other words, it cooks the meat in a very rapid and a very small location. And then, if you notice, that is surrounded by a round tube that will collect the vapors that are given off when this device is applied to the muscle. And it's the evaluation of those vapors which tell you something about the product. And so here is an example. You would have the eye knife. It would be applied to the surface. So you would quickly get that heat on the surface, get some volatiles created. Those volatiles would be drawn up into an inlet tube, which can be as long as 3 meters. So you could set the device in a plant and then reach out and cap the carcasses as they go by. It will then go down, get submitted to the mass spec. As it goes through, it is separated. It reaches a heat impactor where it's converted to ions, and some of the contaminants can be removed, and then it will go to the detector. And so this technology is able to detect those aerosols or volatile compounds over a broad range of mass, a broad variety of sizes, enabling detection of lipids and fatty acids that are directly related to meat flavor. In addition to that, it looks like it's very successful in relating with flavor and sensory analysis. I'll show you a few data slides. no sample prep, you can immediately apply that to a steak and get an answer right away. And it's reflective of cooking, right? Because it's heated when you draw the sample. So it mimics what happens when you cook and serve that particular steak. So I have just two quick samples, and then we're going to wrap up here. One of their studies was to evaluate the characteristics flavors of sheep meat. As you see the little diagrams at the bottom here, this is similar to what they think about if you're evaluating beer for hoppiness and maltiness and alcohol content, or even if you're evaluating different kinds of coffee, you get different flavor notes and characteristics. And they deemed in their study that looking at a variety of different lamb, they could identify lambs that had bold flavor, medium flavor, and mild flavor. And using that volatile organic compound signature, that VOC signature, they attempted to measure a variety of lamb carcasses and identify which of the three categories of flavor that those lambs should be placed. This is just an example of part of their output. You can see they can identify very rapidly, in a matter of just a few seconds, after that eye knife has been applied, they can give answers as to what the characteristics are in that sample. It's really remarkable, I think, really amazing. And here's the example. And it was all done on one population, but on that population, you can see they were 100% successful at identifying lambs that should be in the bold or the medium or the mild flavor character. That is really exciting and promising results on this technology. And you can see it took some sophisticated statistical analysis to be able to do that. I neither understand it nor will attempt to try and explain that today, but Dr. Warner would be happy to explain that to you, but highly successful. Then they did another study, the same kind of thing, where in this case they were trying to characterize beef, see if you could figure out what kind of beef you're looking at. And so they had 7 different types. They had select, they had low choice, upper 2/3 choice, prime, a dark cutter, which you can see had a wide variety of marbling scores, grass-fed, again, wide variety, and then they had some very high marbled Wagyu cattle as well. And you can see on the carcass, they took a sample, and from that sample, they were able to get steaks to do their testing with. And over the course of their analysis, they ended up with 5 groups. They grouped the select and low choice together, the top choice and prime together, and then the others. And I'll show you a couple of data slides. They're all the same. The important thing to look at is here. If you look at precision, they were 100% successful at identifying Wagyu, grass-fed, and dark cutting beef. 88% at identifying top choice and prime, 73% in choice. So overall, 87% successful at identifying the type of carcass from that REIMS analysis. In terms of sensory classification, they divided the samples into positive, neutral, and negative sensory reactions. And you can see they were 60% successful in classifying the carcasses in the study in that way. When they looked at slice shear force, about 75% success at identifying 2 classifications, either tender or tough. They also have additional studies, which Dr. Warner summarized, and I will not summarize today, looking at beef briskets, looking at lipid stability or T-bars, oxidation, on some Australian beef pastrami, and then predicting flavors differentiating depending on how products were cooked in products. So at the end of the day, then, what I would say is that there are a variety of things that impact what it is that we look at when we look at physicochemical and sensory properties of meat. And I think the most important of those are color, marbling, tenderness, and flavor. As we've talked about, I think you have to be careful how you measure and how you report color. It's clear that consumers can tell differences in flavor and they're willing to pay for those differences. That marbling is related to both flavor and tenderness, and that tenderness is pretty complex. It's a challenge. Flavor is too, of course. Multispectral imaging system has been developed for prediction of tenderness, and the REIMS system looks like a very promising technology under development to classify particularly meat flavor. So we've been going for an hour now. That's probably longer than I should have gone, but a lot of good information I wanted to share with you. And with that, I will now stop and be happy to respond to questions if you have any. Thank you very much for your attention.
So thank you, Dr. Calkins. So on behalf of everyone here, I would like really to thank you for this amazing presentation. It was so perfect. It was with a lot of details, and I'm pretty sure that we'll have some questions to you. So let's see. So here we go. We have one. So great presentation, Dr. Calkins. Could you tell about why the Warner-Bratzler shear force is most of the time evaluated in longissimus dorsis muscle? What is the relation with other muscles?
Dr. Flanzer, hello. Nice to see that you're on today. Yes, well, shear force is most often applied to the longissimus muscle. It's the largest one that has that's tender, and so has most of the economic value. It's also really easy, as you know, to access compared to many of the other muscles. And so just almost tradition alone says that that's the muscle to use. You can certainly do Warner-Bratzler shear in other muscles. That's been widely done, of course. But the relationship of absolute shear force value of one muscle to another is not a great relationship. And so we sometimes use a cutoff value for tenderness of the longissimus muscle, and then we know that there are a family of other muscles that are usually equal or superior in shear force to that muscle. But to actually take a shear force of a different muscle and try and predict shear force of the longissimus muscle, there's just too many differences in connective tissue, in all of those other muscle fiber characteristics and fatness that we talked about, that those relationships are modest at best. They're not very high.
Okay, thank you. So the other question is 36 months of age for steers could be considered a young animal when talking about collagen?
Yeah, so collagen, or the connective tissue, that changes. Every month that the animal gets older, the connective tissue or collagen changes as well. And so in our laboratory, for example, we've measured tenderness differences between animals that are 14 months of age and animals that are 20 months of age because of the collagen or connective tissue. So 36 for us is our limit in terms of what we would still consider a young animal. I can tell you that in the United States, the average is far closer to 20 or 21 months. But even if you went from 21 to 30 months, you would see a difference in collagen solubility in those animals as well.
Okay, that's good. So great presentation, Dr. Calkins. I stake with pHU of 5.9 is more related to a normal or a DFD stake?
Yeah, 5.9. Well, you're in kind of a gray area, right? Because dark cutting, or DFD, is a continuum, and in the most severe form, you can get very high in the pH 6.8, 6.9, sometimes even 7 in extreme cases. But more often what we see is not the most severe kind of dark cutting, but something that's more intermediate. Okay. And that typically is in the range of the low 6s. 5.9 is right on the edge. We'd expect typical beef to be about 5.6 or 5.7. Just a word about pH measurement. There are there are a lot of different ways that we use to measure pH measurement. And using a sphere electrode will give you one value. If you homogenize in liquid, the ionic strength of the liquid will change a little bit because you usually just use water, and consequently the pH can change slightly. So be very careful when you're comparing a pH measurement from one study to the other that you look at how the pH was actually measured, because that could have an impact. So I would say a pH 5.9 is tending toward the dark DFD side, but it's certainly not a severe case.
Okay, that's nice. So do you believe that it's necessary to segregate even more these classes, like an atypical DFD?
In most of the time, we have enough variation in eating quality because there are differences in muscles, differences in maturity, differences in marbling, all the rest of that, that a subtle problem with the DFD is really not too concerning for me. But once it gets significant, then I think we need to absolutely differentiate those carcasses. So for me, most critical, those significant DFD carcasses need to be isolated and separated. If you're going to try and market a premium product or a product that you're going to hold up as being especially great from a sensory standpoint, then I would probably get those intermediate carcasses out of the system as well. But it's not as crucial as the big ones. The darker the meat, the bigger the problem, particularly in terms of flavor.
Okay, that's good. So, and do you believe that the consumers can identify differences between a normal and a DFD steak beyond the color, what is evident?
100% they can tell a difference. It's really an obvious. Here at the university, I think once a month or more, someone will call us and say, I have some meat that tastes funny, what is it? And almost without testing, we know that it's got a pH difference. And so consumers are very sensitive to that flavor difference.
Okay. So what would happen with a small piece of cauterized meat for the REIMS evaluation? Would it be cut off or discarded after the evaluation? Does it characterize an invasive analysis?
Yeah, that's, I think personally, you just get my personal opinion on this. Yes, I think it is an invasive analysis. But if you saw in that diagram or that picture, it's not a very deep cut. And so if you could classify the carcass and only lose 10 or 20 grams of meat in doing that, that's a pretty minor cost to pay. Nevertheless, if you were to ask me, would you call this invasive? I would say yes, by my definition it is. I'm sure it would have to be cut off before it could be sold. Okay.
So if tenderness is the biggest factor influencing satisfaction, do you think hump height should be used in the carcass evaluation process?
I answered this question one time in Mexico, and I had someone yell at me by the time we were done. So it's not a very popular answer. But I would answer it this way, Dr. Brici. In the United States, hump height is indicative of the amount of zebu or Brahman breeding in animals. Purebred animals, of course, will have very high hump. Half-bloods will have a smaller height. A quarter will have even less than that. And then those without any zebu breeding, the hump is fairly small. And so in our system, we can use hump height as a way to remove a Brahman or zebu breeding from certain brands or classes of cattle. And it is done in specialty programs and branded programs. Traditionally, if it's just commodity beef that you buy in the grocery store, then actually we would probably not differentiate based on hump height. It would just get mixed in and contribute to the variation that we see. And of course, I would also remind everybody that the reason why there's some discrimination against zebu breeding, at least in the United States, is that genetically, there are some differences in that calpain system, in that proteolytic system between the enzymes and the inhibitors, which are just genetically different between zebu and Bos taurus and Bos indicus breeds. And consequently, that's why bias is there. So I guess I'm thinking about, in particular, a Brazil application. If hump height helps you differentiate between full bloods, 3/4, half, and quarter bloods, yes, I would expect that that would be a useful distinction to make. But if you're trying to say an animal with a large hump versus one that's a little bit smaller and the difference therefore is not percentage of zebu breeding, but perhaps genetics, that's an entirely different question. I know of no data to answer that. It'd be an interesting thing to explore.
Okay, that's great. So, hello, Dr. Calkins. Great to hear you. Very nice lecture and greatly appreciated. Thank you. But so just sorry, my bad. So we don't have any more questions. So we are going to start with the comments. But before that, I'm going to ask my question that I was attending the presentation, and I was thinking about the application of this process to measure, to predict the tenderness. So you talk about the near infrared, and I would like to know is there in the United States some industries that have already adopted this, and how it could impact on the price for the consumers?
That's a really good question, and actually it's far more.
Just a minute, I think we have a problem. Hold on, don't move. We are going back. I just want to say, don't move. We are going to try back with Dr. Calkins, and he's going to answer the final questions, and we are going to finish. Hello, Dr. Calkins. Yes, I can hear you. I can hear. Okay. So, hello, everybody. We had a little problem. The computer of Dr. Calkins just shut down. So he's going to reconnect it with us. So please wait 5 to 10 minutes and he will be back to answer the final question. And if you have any questions, you can do the questions. You can have a few minutes just to think about it. So we are going to be here for 5 to 10 minutes. So I don't know what we can do, but I'm going to take this time also to thank you to the attendance for your involvement, for the question. It was a great pleasure to be here with you with this sharing, it's a really great to be here. Eu vou aproveitar que o Dr. Calkins não está online também e vou dizer para vocês que se vocês quiserem fazer alguma pergunta em português, fiquem à vontade, a pergunta pode ser feita em português também, tá? E como eu expliquei agora, o Dr. Calkins teve um problema técnico, o computador dele desligou e ele me ligou e falou que ele vai precisar entre 5 e 10 minutos para retornar. Então, eu gostaria de pedir, por favor, para que vocês continuassem online e esperassem para que ele possa voltar e responder a questão, e também para finalizar, para que ele possa ouvir os bons comentários que todo mundo está fazendo sobre a palestra dele, tá bom? Eu agradeço muito. Apenas um minuto eu vou falar com a organização, o comitê de organização, só para ver a quantas nós estamos. Bom, enquanto isso, vamos fazer um bate-papo. Eu gostaria de saber de onde vocês estão, onde tem gente assistindo a gente. Eu vi que tem gente de Cascavel, eu vi também que tem gente da Colômbia e bastante gente de Londrina. Da onde mais? Tem gente de Campinas também, se eu não me engano. Ah, olha, a Carolina está falando, gente. Estou falando com o meu comitê de organização, que ela fez uma pergunta e que não anexou. Se vocês puderem dar uma verificada. Carolina, se você puder reenviar, a gente agradece. Ó, tem gente de Foz do Iguaçu, do Paraná, gente da UEM, de Goiânia, que legal. Que legal, gente. Tem gente da PUC de Curitiba. Muito legal. Tem gente de todo lugar do Brasil. Da Exalc, de Piracicaba, muito legal. São Carlos, a Renata. Oi, Renata, bom ter você aqui com a gente. Da Embrapa São Carlos, né, Renata? Gente, eu acredito que o Dr. Calkins ele já deve estar voltando. Eu acredito que tenha acabado a bateria do computador dele. Obrigada, Carolina. Gente, a Carolina mandou a questão dela, então eu acho que a gente pode também já deixar no stand-by. De Volta Redonda, Rio de Janeiro, que legal. Da UFF. O UFF é a Universidade Federal de... Gente, da UEL. Uma vez o UEL, sempre o UEL. Saudades do calçadão. Sorry, guys, we are back with Dr. Calkins. Hello, Dr. Calkins. Dr. Calkins?
Yes.
We are hearing you, so that's good to have you back.
On a poor connection until my computer boots up, so I thought I would try again here.
No, that's perfect. Universidade Federal Fluminense, que legal. Gente, eu acho que o Dr. Calkins caiu de novo, ele vai reconectar. Nós temos também gente de Washington, que legal. So, Dr. Calkins, we are back now. We're still live. So I'm going to give you the time to answer the question that I give. And in time that we were waiting for you, so we had some more questions, which is really good. So I will let you the time to answer the question. So I have asked if this new technology, the near-infrared technology, used to predict the tenderness of meat on slaughter plants, if it will have a cost to the consumers, and if there's some plant in the United States that have adopted this technology. Dr. Calkins? I think Dr. Calkins is not hearing me. You are hearing me? Okay, that's good.
I will try to answer. The connection is terrible. I apologize.
Okay.
The system is just been developed and is currently available, but it's not far enough along to have been implemented into the U.S. industry just yet. But
Oh, guys, I think that we will not be able to finish today with Dr. Calkins because his connection is really poor today. He's just telling me. So I'm really, really sorry. But we had his presentation, which was a really good presentation, and we were able to go until almost the finished question. So sorry, Carolina, you stay without your answer, but I really thank you everybody for being here with us and for holding on, waiting for him to come back. Eu vou falar um pouco em português também. Eu agradeço todo mundo que esteve aqui conosco durante esse webinar, que ficou aqui agora aguardando o Dr. Calkins voltar. Infelizmente, eu acho que não vai ser possível finalizar esse round de questões, mas muito obrigada a todos, muito obrigada por estarem conosco, muito obrigada por essa troca de informações, por essa troca de ideias. Foi muito legal e eu vejo vocês no próximo webinar.